Thanks again for the introduction, Brett. And when Greg asked me to talk about this, I went back a few years, in the distant past and looked that I gave similar talk in one of the symposium, and I focused on innate immunity, and that's still relevant. So I'm going to start by maybe a refresher of immunology for you all, at least to get back on track. I know many in the audience don't necessarily have the deep background in immunology, so you got to bear with me. And we'll start with this schematic, if I can find the pointer, of the chicken and different lymphoid aggregates from the avian immunology book. I highlighted the thymus, which is down here. You got about 7, 8 lobes on each side, and the bursa. Everybody knows that. And these are the primary, what we call them primary immune organs. T cell development takes place in the thymus. B cell development takes place in the bursa, and those are the antibody-producing cells. But our focus in this symposium is going to be in this whole area. That's the small intestine and the ceca. And the rest of the talks are going to be addressing all those infections. That being said, this is a little bit of a schematic reminding everybody what the immune system, how it works. We have 2 branches, what we call innate immunity and adaptive immunity, or acquired immunity. And innate immunity basically kicks in right away. Just kind of like when you get bit by a mosquito, all of a sudden you get inflammation and redness and all of that, and infiltration of cells. And that involves a bunch of mediators and phagocytes, also complement system, and what have you. On the adaptive or acquired immunity side, we have 2 main branches. The B cells will produce antibodies, and the T cells will basically deal with intracellular pathogens, and I have a couple of slides on that later on. So, starting with innate immunity, the system possesses what we call a limited number of pathogen recognition receptors. These are unique receptors on the innate immune cells that recognize specific ligands on a whole host of microbes. And what we call those are PAMPs, pathogen-associated molecular patterns, or MAMPs, microbe-associated molecular patterns. And one thing very important in the innate immune system or response is there's no memory generator. So basically it's like a memory of a fish. You get 5, 6 seconds, and then after that you forget, and you attack the same pathogen, the same response all over again. But these things will limit the capacity of the innate immune system to deal with evolving pathogens, so to speak, or with pathogens just like Eimeria that has several stages at intracellular and extracellular to deal with. Yet it's very immediate. It helps slow things down and bridge that response to allow the acquired immunity to come in and take care of whatever insult you have. So if it's so limiting, why is it interesting and worth looking at? It's because the innate response deals with the vast majority of insults and pathogens and potential infections. And we usually don't see that because they're stopped in their tracks by the innate immune system. So oftentimes, I tell my students, I'll be lecturing, like, all of us in this room are dealing with something right now, but we're not sick, we're not infected. That's because the innate immunity is dealing with it. But what's more important is, and for the longest time, we were under the impression that these are 2 different or separate entities. They act in basically independent fashions, and that's far from the truth. They act together, innate and adaptive, all the time. So there's always like a loop that feeds each other. And it initiates and shapes the adaptive immune response, and I'll show you in a bit in a couple of examples. So what also happens is adaptive immunity uses these cells of the innate immune system as important effectors in acquired immune responses. And I have an example here. If you have an acute infection, you immediately have that innate immune response. You got antimicrobial activities, all sorts of stuff gets secreted, cytokine releases. Some of those will actually trigger the adaptive immune system. That means that's when the innate immunity is calling for help. I can't deal with the insult by myself, come help me. So there are distinct cell populations for each innate and adaptive. However, as I mentioned earlier, they actually work together. We just have them classified as such because they are the first responders. And the ones that respond the first are heterophils. So we don't have a whole lot of populations of heterophils in remote sites, but they're always there, and there is immediate call for migration of additional helpfils, heterophils and other immune cells. In other words, you have that infiltration trickle down systemically to the site of infection. Monocytes, as they majority will basically develop into macrophages and also dendritic cells, and these two we call professional antigen presenting cells. So whenever you have intercellular infections, just like Eimeria, these cells will process the antigen inside, and they will present it on their surface for other immune cells, the adaptive immune response, specifically T cells, to act upon and be activated and trigger a cascade of the response that's needed. Mast cells also just, they're granulocytes as well. They use a bunch of granzymes to kill mostly bacteria, but also they can deal and neutralize larger pathogens. And we also have the complement system. And much of the processes that T cells undergo is phagocytosis, antigen presentation, as I mentioned. And in case of heterophils, but mostly you have the oxidative burst. So the heterophils will take a bunch of cells or pathogens or microbes inside, and they burst and kill them, killing themselves in the process. But most importantly, they secrete a bunch of cytokines and chemokines and all other antimicrobial peptides like defensins. So here's a list of the two main things that we deal with in terms of the innate immune response. And we have the soluble portion and the cellular portion, and here's an example or a list of it. And when you're all doing research and looking at particular parameters to measure, you'll probably be looking at some of these. So we have host defense peptides or antimicrobial peptides, lysozymes, other enzymes, acute phase proteins, also the 3 complement system, although it's not very relevant when you're dealing with coccidiosis. And these are the cells, and I've listed a few examples here of, does this work? I don't know if this is working anymore. Oh, it is. Can't see it from this angle. The key inflammatory cytokines that are secreted by heterophils, which provides not only a loop for more heterophils to come in and do their thing and come for the help, but also they start triggering the acquired immune response. Yeah, we'd have to plug it in first. Okay, we'll do it at the end. Thanks. Same thing for macrophages. Some of these are interleukin-1, TNF-alpha, and other cytokines that, just like I said, they act in autocrine fashion, so they self-promote themselves, their proliferation and activation. Dendritic cells also that secrete IL-2, IL-23, as well as IL-12, and those are key cytokines that will help activate T cells. Natural killer cells, that the name indicate, they basically non-specific in a way, yet they are specific because they can tell there's something wrong on the surface of an infected cell or cancer cell, something abnormal, doesn't have the correct receptors, so they automatically and indiscriminately attack it and kill it. So here are the arms, the adaptive immune response, and we have basically two strategies, humoral and cellular. Humoral is basically involves the B cells and production of antibodies, so anything that's secreted. Cellular immunity is mainly mediated by T cells, but it also involves B cells. So everything is at the same time happening together, but in different ways. So the receptors, in this case, in B cells, you have a bunch of receptors on the surface, and these are the immunoglobulins or the antibodies, but they are surface antibodies. Once the B cell differentiates and starts secreting antibodies, Those are the highly specific antibodies that will neutralize a virus, or a sporozoid, or what have you. And receptors of the T cells, they have quite a different function. They're not secreted. They just recognize antigens in association with the antigen-presenting cells like the dendritic cells and macrophages. And here's some of the examples what the effector functions are, just for your refresher, so to speak, in terms of humoral. You got the B cells here producing antibodies, and mostly what they do, because they're secreted outside, they act on extracellular pathogens, and while cell-mediated acts on intracellular pathogens. So most important for us in this context is mucosal immunity. As we all know, the mucosal surfaces basically are the major route that most of pathogens gain access to the host, to the bird or to us or whatever. And digestive tract obviously is the main route for that. And it has a bunch of non-immunological, immunological defenses. And as example, you have the epithelial lining is very important. You have the mucus layer right on top of it. And also we have the commensal microbiotas. They can produce bacteriocins and things like that that are antimicrobial. pH, low pH and high temperature of the bird helps. Most importantly is the gut-associated lymphoid tissue, and that's the GALT. So immune cell populations are quite diverse. You find them in the gut at different proportions based on where they are, based on the situation, what's going on in the gut itself. And a lot of external factors play into that. So when you're running trials, pay attention to everything on the right side here, because differences on those external factors will influence the immune response. So you have to keep that in context. Here's some of the examples of the epithelial lining, which is very important, how they work. And they themselves, they're not necessarily considered immune cells, but they produce a bunch of host defense peptides. These are the ones that most of you, when you're running trials, you're measuring these through qPCR mostly, and also the enzymes and other factors. All right, so why am I telling you all of this? To get to the point, because as Dr. Cervantes just explained, this is just a different depiction of what the cycle is, and this is a picture of a sporozoid, and they use the apical surface to enter the epithelial cells. Before then, they're external. Once they go in and they start the asexual reproductive cycle, they're internal. So what's happening with the immune system is trying to react to every single stage, right? So it deals with a bunch of stuff, but when it comes to coccidiosis, you're looking at humoral immunity. Yes, there are specific antibodies to the sporozoid as well as the metazoid stages of Eimeria. They're not, though, very effective in protecting because it's a very short time frame. And at the same time, when you have external sporozoids and metazoids, you have other stages going on inside the cell, and they're destroying new cells. So the T helper cells are the main cellular immunity, cell-mediated immunity, or CMI, players here. But we also have cytotoxic T cells. They act like natural killer cells, and Tregs. And as Dr. Cervantes said, it takes a while for the full cycle to go, and it's self-limiting in the fact that it doesn't want to destroy the host, but then also immunity builds up, and the host has that immunity to deal with it. Just to give you another example of what all these mean, and I'm not listing anything, specifically in detail. I'm just giving some example. There are a whole host, an array of cytokines and chemokines and transcription factors with every single stage. But to give you an example, with the dendritic cells, NK cells, producing these key factors that will trigger the T cell activation to go and do its job. And you have three main T cell subpopulations, so to speak: T helper 1, T helper 2, T helper 17. And on that note, those T cells, in turn, they produce key cytokines. The key one that everybody measures when you're looking at coccidiosis is interferon gamma. Why? Because interferon gamma is produced mostly by T cells, by NK cells, and it's the main factor that triggers macrophages. Macrophages need interferon gamma to act, to become activated. So there's a whole host of different loops such as this that communicate between the innate and adaptive responses. So it was very difficult reading the most recent literature that tried to summarize that in one slide, but I tried my best. What most everybody does, for good reason, based on what I just presented, you have mostly inflammatory markers, right? So interleukin-1, 6, tumor necrosis factor alpha, and those are part of the innate immune response as well as adaptive immune response, although we call them inflammatory responses. But as I mentioned, T cells will promote macrophage activation and all of that that entails after activation. You have innate immunity. Most of us look at host defense peptides. The most that we deal with in response to cocci are the beta defensins, but also what we call LEAP2, liver-expressed antimicrobial peptide 2, even though it's found in the liver initially, but it's highly abundant in intestinal tissues. Key adaptive immune elements, interferon gamma, IL-12, as I mentioned, most everybody looks at that, so we're all on the right track. Also, we look at pathogen recognition receptors and often focus on TLRs, toll-like receptors. These are the most commonly known or studied. We don't know a whole lot about other receptors, although they are very important in the inflammasome, and that's something we rarely look at in the context of coccidiosis, things that need to be considered. Gut integrity component, many of us look at tight junctions, but how is that relevant when the parasite itself is destroying the epithelial, the enterocytes? So of course you don't have tight junctions anymore. So it's kind of tricky looking at all of these in the context that you're looking at. So nutrient transporter, obviously, one of the phenotypes that we look at is, like Dr. Cervantes said, body weight gain, and you have also FCR. All of those are impacted. So we try to correlate all of that during coccidiosis. Immunometabolism, cellular oxidative stress, and just a couple more that are not up here. We don't look at them, up closely, so to speak, but it's always very important to put everything in context of the design of your study. And that's the challenging part, that we don't always get repeatable results because of all these factors that we see. And I guarantee you, you don't have to look at what Dr. Cervantes or Dr. Jenkins did. Look at your own trials, and you're going to see different results every time, because any of those factors that you didn't take into consideration will affect it. So it's very important to put everything in context. So speaking of that, we went to a field study. We collected samples by we, I mean my students. They're sitting in the back somewhere. They do all the work. We take all the credit. We went to farms that are considered low performing or high performing, and collected 16 samples, 8 males and 8 females, or 8 birds. Did lesion scorings. These farms had a history of exposure to coccidiosis, just like almost every other farm in the US. And we did RNA-seq, and our collaborators, I don't know if Dr. Romero is here in the room, he did a really nice job coming up with a new quantitative pathway activation method to assess how these two entities, based on performance, so putting it in context of performance. So we have low performance and high performance, and the green ones are the ones are activated in the low performance versus the high performance. The same one for here, but these are kind of like lower expressed. And obviously, you can't read most of these, so I blew them up. The main categories were looked at: nutrient metabolism, immune function, gut integrity, oxidative balance, digestion, absorption. And as you can see, most of them, a third at least of these are in the top category. But the second biggest part is immune function. So it goes to show how much it goes on. And to summarize that simply is we found activation of immune response pathways, gut integrity in terms of cell replication because of more sloughing, I guess, with those flocks, oxidative stress, and several amino acid metabolism. These were basically greater abundance in the low versus the high. And on the opposite side, pathways related to digestion, absorption, vitamins, minerals, and what have you, they were actually lower in the low performance. So put that all in picture is resource allocation aspects and things like that. You can kind of conclude that based on this, that's why we have low performers, high performers, but obviously we don't know when the management is. I'll get 4 minutes, so I better rush. All right, so other things that we're trying to use to get better idea in a more controlled environment, we started using iohexol. This is a big molecule used in imaging and in medicine. It's much more stable. I'm sure most of you, if not all of you, have heard FIT-C, dextran. This is much more stable. And we did a small study with our necrotic enteritis model. We had no infection, Eimeria maxima inoculation, Clostridium alone, and Eimeria and Clostridium. So we induced very subclinical, barely any lesions. We saw some lesions, but we had no mortality with these birds. But we measured these at day 21, which is basically the typical peak necrotic enteritis. You see the huge difference in the iohexol. And this is showing that you have breakup in the gut lining, and things are going through between the cells, through intercellular space, into the blood. And once you add EM to the picture, you even have greater value there, but there were not much difference. But it goes to show how much EM alone does and how it influences the gut. And when we looked at some of the key cytokines, before I get to that, day 28, a week later, we see no differences. So everything went back to homeostasis type format. We figured the birds have recovered. And we look at some of the key cytokines, the same story. We look at IL-1 beta, interferon gamma, and IL-22, which is involved in homeostasis. Day 21, they're all high up with the inoculated, or dual inoculation or EM only, but nothing on day 28. Now, continuing on, very quickly here, I'm going to run through something very important it's often understudied, and that's the evasion of immunity by pathogens. And they have several ways to do that, especially coccidiosis and Eimeria parasites. And one we dealt with a little bit is mimic the host cell molecules or inhibit the immune response and things like that. So cytokines are actually also produced by these parasites. One of them is called macrophage migration inhibitory factor. And the USDA group, Dr. Jenkins and his group years ago, they isolated or characterized the cytokine in Eimeria, both Tenella and Iservalina. We followed up with studies. We also looked at the turkey myth. And following that, we wanted to see whether or not they act the same way. And sure enough, in addition to a major receptor that's called CD74, these Eimeria myth use the same CXCR4 receptor or the complex receptor for both the chicken myth and the Eimeria myth. And so for that, we decided, okay, so this is what's happening here. They're acting the same way as the chicken myth itself. And what happens is they're doing this because they inhibit the migration of macrophages, and thus they reduce that. Oh, my time's up, almost a minute. Okay. Okay. Oh, 2 minutes? Okay. So, thank you. So what we decided to do is, okay, so what happens if we knock this out or knock it down? Does that change things? So we designed a study with CRISPR-Cas9, quickly I'm not going through details of this, to look if we get MIF deficiency in Eimeriya tenella, how does that impact the response of the host? And following that, we found there is a diminished MIF expression in the transgenic parasites after generating that, and that caused a tenfold reduction in the Eimeriya MIF that's produced by these transgenic Eimeriya. And functionally, what did that mean? It actually resulted in almost 14% increase in live cells and livability of cells. So it reduced the effect of Eimeriya. So what that means is, again, these parasites will secrete, will produce things to help themselves, and it's an area that we don't look into enough. It could be providing certain ideas, at least, or potential ideas to develop vaccines in that context. Last but not least, if you all went to my student's presentation on Monday, she did a fantastic job looking at a mini gut model. She developed all of this herself, taking embryos, taking intestinal sections from the embryo, culturing them, and building enteroids, basically what we call apical out. Okay? And she proved that they can actually develop in vitro, just floating. And to apply that, she did infect them with Tenella sporozoites. And I don't have the fancy 3D videos that she did on Monday. She provided on Monday. But you see the development and the invasion and different developmental stages of the parasite in that. So on that note, here's my summary. I'll go back to it. Let me acknowledge everybody here. My graduate students, I hope they're all here. Well, I know one of them is not here. And Dr. Romero, hopefully he's here, and USDA funding. But here are the summary. So, points. We have to be careful with how we interpret our data. So always contextualize things. I know we have limited toolbox in terms of immunological reagents and such, but we have to work with what we have. And most of us look at qPCR. Keep in mind, that's an indirect measurement. That's only at the RNA level. We don't know what happens at the protein level. With that, I don't know if I have time for questions, Brett?
We're right at it. So.
Okay.